Our next topic for Chapter 11 is neuron structural classification. Structurally, there are three types of neurons: multipolar, bipolar, and unipolar neurons. We've already looked at the functional classification of neurons, and that was a motor neuron, an interneuron, and a sensory neuron, because that is what neurons do. They either act as a sensory, or motor device to pick up on information and convey it not only from the periphery of the body, but to the brain and then back out to the effector cells. But structurally, we're looking at what do neurons look like. They have many processes. Some have 3 or more. That would be a multipolar neuron. Some have 2. That would be a bipolar neuron. And some have just 1. But even unipolar neurons have a single short process that splits into a proximal and distal branch. So it'll have 2 parts, one going to the peripheral nervous system, the other towards the central nervous system. If we were to look at this image in table 11.1, we can see the multipolar neuron on the left. It has many dendrites and 1 axon. Dendrites are still the receptive region, and the axon is the conducting region. The dendrite and axon of the bipolar neuron is going to be, respectively, the receptive and conducting region of that bipolar neuron, but there's just 2 processes or things coming off of that type of neuron. We can see for the unipolar neuron, also called a pseudo-unipolar neuron, that it has only 1 process that directly comes off the cell body, but that single process immediately splits into the peripheral and central process. The peripheral process is receptive, whereas the end of the central process is going to be the end of the conducting region and will be carrying signals towards the central nervous system. There's different structures even to our structural classifications. We can have a different arrangement of dendrites, a slightly different diameter of axons, but we want to remember that multipolar neurons are generally going to be motor or interneurons. Bipolar neurons are found only in the special senses. For example, they are the second neuron found in the visual pathway of information going to the brain. And then unipolar neurons are generally going to be sensory as well. So here we can see a comparison of the structural classes of neurons with their function. So again, think multipolar, motor, and then also interneurons, so multipolar, motor. The most common type of structural class for neurons is going to be the multipolar neuron. For bipolar neurons, always think special senses. And for the unipolar neuron, remember it is going to be mainly sensory neurons. The sensory neuron that takes information, say, from the skin to your spinal cord in order to allow that information to ascend to the brain, that would be a sensory neuron that is unipolar. So just a reminder, structurally, the classifications for neurons are unipolar, multipolar, and bipolar. The functional classifications, however, are sensory, or we could say afferent, motor, or we could say efferent, or interneurons. Another name for interneurons are association neurons, because they take the information that they get from sensory neurons, associate it with past experiences or other information coming from other parts of the body, and determine a motor output. Next, we're going to move in and look not just at neurons, not the cells that help hold them together, not at the different parts and how electricity travels through a neuron, but the electrical principles that allow the nervous system to be a communicating system. So I've taken this out of your textbook, so I will read it a little bit verbatim, but the human body is electrically neutral. It has the same number of positive and negative charges. However, there are regions where one type predominates, making those regions positively or negatively charged. What this is saying is that even though the entire human body is electrically neutral, if we were to look at one location or look locally, say at a neuron, we would see that on one side of a cell membrane and the other side, there is a difference of charges. Now, if we took the whole entire human body into account, we would have the same number of positive as negative charges, therefore making the human body neutral. But we're going to be focusing on these single locations, these locations across a barrier, those barriers being plasma membranes. Neuronal transmission relies on these local differences in change. Neuronal transmission reflects the flow of chemical ions in and out of neurons. Even in Chapter 9, we were already acknowledging an influx and efflux of ions such as sodium and potassium and how that could change membrane voltage. Just like in the cell membranes of skeletal muscle cells, we see the same thing occur in the cell membranes of neurons. The influx and efflux of ions will change what is called a resting membrane potential, and changing that resting membrane potential can create an action potential, or an electrical signal that travels down an axon to an axon terminal. A couple of principles of electricity need to be acknowledged: voltage, potential difference, current, and resistance, just so that they're not unfamiliar. So voltage is the measure of potential energy generated by separate charges. So again, these charges are going to be separated by a plasma membrane. The potential difference is the voltage measurement between 2 points. The difference between the voltages on each side of the cell membrane creates a potential difference. And if we change this potential difference, we can introduce or create a current. A current would be a flow of electrical charge from one point to another, so say from the axon hillock to the axon terminals. And if we introduce any resistance, any hindrance to that charge, to that current, we will reduce current and impede the action of that potential reaching the axon terminals. So insulators would offer high resistance to flow. One such insulator is myelin sheath, and that's why in the peripheral nervous system, the location of Schwann cells does not allow current to flow through channels, because the myelin sheath is forming an insulation, and that insulation is a location where there will be no flow difference. Conductors have low resistance, and these would be areas such as the nodes of Ranvier. The nodes of Ranvier do have ion channels that allow influx and efflux of ions, therefore flow. Now, while no math is required, we do want to just briefly mention Ohm's law. Ohm's law is just giving you the relationship between voltage, current, and resistance. We can see the equation is current is equal to voltage over resistance. That means current and voltage are directly proportional. So whatever happens to current, same thing happens to voltage, whereas current and resistance are indirectly or inversely proportional. So whatever happens to current, the opposite happens to resistance. So let's start with that resting membrane potential. So here we have an axon. We're looking at the inside and outside. The inside and outside of the cell is separated by a barrier called the plasma membrane. The inside of a neuron is more negative than the outside of a neuron. That doesn't mean the inside of the neuron is a negative number. It just means that it's going to be more negative or closer to zero than the outside. We will, however, represent resting membrane potential with a negative number just to make the calculation make more sense in our brain. So for example, resting membrane potential, the potential difference across the plasma membrane when a neuron is not firing an action potential is negative 70 millivolts. This is because there is a lot of sodium outside the cell and more potassium inside the cell. And in order to balance out the sodium and potassium levels, we have chloride outside the cell, and we have anions or proteins inside the cell. So we would see the negative and positives balance each other out to produce this resting membrane potential, which is more negative inside the cell and more positive outside the cell. The sodium ATPases, or the channels that establish this electrochemical gradient, were talked about in chapter 3 of your textbook. There will also be leakage channels that allow some potassium to leak out and some sodium to leak in. But if we were to pause the leakage channels and the sodium potassium ATPase, we would see that at any one point in time, a cell's interior would be negative on the inside compared to the outside, which would be slightly more positive. The difference in these positive and negative charges is about negative 70 millivolts. So before we move on, we are establishing that resting membrane potential is negative 70 millivolts. I highly recommend you watch this video. The link is included in the PowerPoint. You can find that on Blackboard. The name of it is the A&P Flicks Neurophysiology Part 1, Resting Membrane Potential. It'll talk about how we establish resting membrane potential you'll see movement of sodium and potassium in and out of the cell membrane, as well as the work of the sodium-potassium ATPase. The chemical ion channels at work, as we talk about forming or creating an action potential, will be a passive or leakage channels, chemically gated or ligand-gated channels, voltage-gated channels, and mechanically gated channels. So the leakage channels are always open, but they only allow very small ions or substances through. Chemically gated channels allow a specific chemical through. We saw chemically gated or ligand-gated channels at the junctional folds of the neuromuscular junction in Chapter 9. Voltage-gated channels open or close in response to a voltage change or a membrane potential. And the mechanically gated channels are physically opened or closed. So they're going to open and close in response to some type of deformation of their receptors. When these channels, whether they're passive, chemically gated, voltage gated, or mechanically gated, when they open, ions are allowed to pass through. If ions go into the cell, that's called influx. If ions leave or exit the cell, that's called efflux. And this movement of ions, which are charged, can change the membrane potential. So movement of charged ions can disrupt the resting membrane potential. And that's what we see occur when an action potential is generated. Let's look at some examples of these specific types of channels. So first we'll look at the sodium-potassium ligand-gated channel. The sodium-potassium ligand-gated channel would open when a neurotransmitter, like acetylcholine, bound to it. A ligand is a chemical that must bind, whether it's binding to an enzyme or a channel, it will be able to perform an action or cause an action when it is on its receptor. When a sodium-potassium ligand-gated channel is closed, sodium or potassium cannot enter or exit. When it opens, we generally see a sodium influx and a potassium efflux. We see both ions enter and exit through the same channel, so this would be an example of an antiporter. For that definition, please see Chapter 3 in your lecture textbook. So here's operation of a ligand-gated channel. If this channel looks familiar, it is also called an acetylcholine receptor, and you saw it on the motor end plate, or specifically the junctional folds at the neuromuscular junction on the sarcolemma. So it would be important then to remember what makes a muscle contract, because at the neuromuscular junction, we saw a ligand-gated channel bind to the neurotransmitter acetylcholine. Acetylcholine binding to its channel opened that channel and allowed sodium to go into the cell, potassium to leave the cell, and the influx and efflux of those charged ions created a voltage change. And we called this a local depolarization or end plate potential. A similar thing can happen to create a graded potential in a neuron, specifically in the area of the dendrites or cell body. So we'll still see the use of voltage-gated sodium channels in neurons. Voltage-gated channels are going to respond to a voltage change. These voltage-gated changes are going to usually be created by first a ligand-gated channel that responds to a chemical, and then that ligand-gated channel will cause a voltage change, thereby opening voltage-gated channels. So it'll be a series of steps that eventually open or cause the operation of a voltage-gated channel. For example, here we have the sodium voltage-gated channel that is closed when the internal or intracellular environment of a neuron is at rest, or when the intracellular environment of a neuron is negative, because the inside of a neuron at rest is generally negative. So sodium can't enter when a neuron is resting, but it would open if the inside of the neuron suddenly became positive. If the voltage or charge of the inside of the neuron suddenly became more positive or less negative, we would see these voltage-gated channels open and further that depolarization or voltage change by allowing an influx of sodium. So in an open sodium voltage-gated channel, sodium would be able to enter the cell. It would cause a further change in voltage away from the negative and towards the positive pole. So this action generally causes depolarization, which is one of the first steps of creating an action potential. Here we can see the operation of a voltage-gated channel. We see that On the left, the channel is closed because the inside of the cell is negative, but when the inside of the cell becomes positive, which is what we see on the right, the channel opens and allows sodium to enter. Adding a positive cation like sodium to the inside of a neuron that's already had a voltage change making the inside positive will just make the inside even more positive, so further generating or propagating this action potential. So before we move on, we just want to establish again that the resting membrane potential is a measurement of the difference between the voltage inside and outside of a neuron. These voltage numbers are separated by the plasma membrane, and we're going to keep them relatively similar by using both leakage channels and sodium-potassium ATPases, or what you may know as the sodium-potassium pump. The leakage channels and the sodium-potassium pump will stabilize the resting membrane potential, maintain that concentration gradient for sodium and potassium by pumping 3 sodium cations out of the cell and 2 potassium cations into the cell. So this establishes a more negative inside to a neuron, establishes that resting membrane potential, and then by utilizing ligand-gated, then voltage-gated channels, we'll be able to change this and create first a graded potential, then an action potential. So changes in membrane potential are what create the transmission signal that we are calling an action potential. The first change will produce a really low grade, or you could call it a short grade type of potential, and that's generally just called a graded potential. Graded potentials occur in dendrites and somas, or cell bodies, and they're incoming signals that are going to travel towards the axon, but you would not see a graded potential in the axon itself. So usually they're only along the dendrites and soma. They are short distance signals, and they operate as incoming signals that, if strong enough, can cause an action potential. If graded potentials aren't strong enough, they don't cause an action potential, and action potentials are all or nothing signals. They either occur or they don't. They're long distance signals that have the same amplitude of a 100 millivolt change in membrane potential. So neurons are going to use these changes in their resting potential as communication signals for receiving, integrating, and then sending information along to the axon terminals. So let's look closer at the 2 types of potential changes. Graded potentials, again, short-lived changes. They belong in the dendrites, belong in the soma. They do not belong or are characterized by going through an axon. They are going to be a current that decreases in magnitude, so it may start out strong near the dendrites, but without any regeneration throughout the dendrites and the soma, it'll die out if it's a weak signal. So you need a strong graded potential in order to cause an action potential. These short-lived localized signals can either be depolarizations or hyperpolarizations. Depolarizations excite the neuron membrane and can generally lead to, if strong enough, an action potential. Hyperpolarizations are going to inhibit or shut down the neuronal membrane, and they generally will not cause an action potential. So graded potentials vary in magnitude. Their voltage varies with stimulus strength, but we need graded potentials. They're essential in initiating action potentials. Action potentials are our long-distance signals. They travel along the axon. They can travel to any excitable membrane, which of course includes nerve cells and muscle cells. Like in Chapter 9, we talked about skeletal muscle being able to be excited by a motor neuron. And not only are action potentials able to be generated in an axon, they're able to propagate or be regenerated along the axon in order for that signal to reach the axon terminal. The most clinical definition of an action potential is a brief reversal of membrane potential with a total amplitude or change in voltage of 100 millivolts. So unlike graded potentials, action potentials being all or nothing means they do not decrease or change their strength with distance. So graded potentials have or show decreasing strength with distance. Action potentials do not. They maintain the same strength with distance of 100 millivolt change in membrane potential. So membrane changes relative to resting membrane potential include depolarization and repolarization, as well as hyperpolarization. So these are the 3 steps of an action potential. An action potential begins when we have depolarization away from the negative pole. We reset the membrane with repolarization back towards the negative pole. And then hyperpolarization occurs because the potassium channels close slowly and slightly polarize the membrane past the original negative pole. So we get a hyperpolarization back towards that negative pole, which is reset by sodium-potassium ATPases. You may abbreviate action potentials as AP, but they're also known as a nerve impulse. And we only get an action potential if an impulse, like the graded potential, has adequately stimulated the axon. Because again, an action potential is an all or nothing signal. It doesn't occur at a weakened state through an axon. It occurs or it doesn't. Always at the same strength of a change of 100 millivolts. Here we have the steps of an action potential. Again, we see we begin at resting state. Resting state here is negative 70 millivolts. When a graded depolarization is strong enough to change the membrane potential of the dendrites and soma to negative 55 or move the negative membrane away from the negative pole towards negative 55, then we've reached threshold. So we're going to set threshold at about negative 55 millivolts. And if we can make the inside of the neuron more positive and reach threshold, then an action potential can occur. And along the axon, we will open sodium-gated channels, allow an influx of sodium, which depolarizes the membrane further because we add more positive to the inside of the neuron. And that will be the first step of our action potential. That'll occur all along the axon. And once the action potential has reached the axon terminals, we will see that the entire axon has reset because the action potential occurs in segments of the axon. So we will see successive depolarization, then repolarization, then hyperpolarization all along that axon. We see hyperpolarization here is below negative 70. So it is below the original resting membrane, but quickly goes back to the resting membrane potential of negative 70 millivolts. And that is because of the sodium-potassium ATPase and leakage channels. So that's a lot of terminology to introduce, even terminology we haven't introduced to understand the action potential. So if some of this isn't making sense yet, now we're going to combine all of the information about not only what an action potential is or what a graded potential is. We're going to combine that with different types of channels and the cations moving back and forth. So we're going to put it all together. So definitely make sure that you're restudying or looking through these slides. So step 1 in generating an action potential or trying to generate an action potential is to start with a graded depolarization. So a graded potential is required to create an action potential. A graded depolarization uses ligand-gated sodium channels, and when a neurotransmitter binds to these ligand-gated sodium channels, they open and allow an influx of sodium. This influx of positive cations will change the negative voltage inside the cell to something a little less negative. So we'll see a change in membrane potential from a negative 70 millivolt potential to negative 55 millivolt potential. If we can do this, we can reach threshold. Threshold is required before a graded depolarization can cause an action potential. The action potential would begin if we had a strong enough graded potential or graded depolarization that reached threshold and then allowed the first part of the axon hillock, to open its voltage-gated sodium channels. When the voltage-gated sodium channels of the axon hillock open, we would see an influx of sodium. This influx of sodium adds even more positive charge to the inside of the membrane, moving us even further away from that negative pole. This would continue to happen all along the axon. We would have segments of the axon depolarizing, repolarizing, and hyperpolarizing in front of adjacent or segments further down the line. As these segments depolarize, they would be reaching a more positive pole, eventually all the way up to positive 30 millivolts. The repolarizing step resets the membrane. We're going to see the inside of the membrane become more negative again when the sodium voltage-gated channels close and potassium voltage-gated channels open. Whereas sodium entered when the sodium voltage-gated channels open, we're going to see potassium exit when the potassium voltage-gated channels open. So if at first during depolarization we added positive to the inside of the cell so that that negative potential became more positive or moved away from the negative pole, now we're going to close those channels and open potassium channels that cause potassium or a positive cation to leave. And as we lose positive ions or positive charge from the inside of the cell membrane, the inside of the cell will become negative once again. It'll go back to the negative pole of negative 70 millivolts. This will restore the resting electrical conditions of the neuron, but not the ionic conditions. The ionic conditions are going to be restored by the sodium-potassium pump or ATPase. Hyperpolarization occurs because the potassium voltage-gated channels are slow closers. What it means to be slow closers is they allow the efflux of even more positive ions than is needed to return back to resting state. So we have a momentary hyperpolarization, or the inside of the membrane becomes more negative than negative 70 millivolts. But again, this is going to be restored because the ionic redistribution that can be accomplished by the sodium-potassium ATPase. So that pump is responsible for restoring the ionic conditions after hyperpolarization. So hyperpolarization is just when the inside of the cell membrane becomes more negative than the normal resting potential. So let's just take a minute and talk about action potentials. We will look again at depolarization, repolarization, and hyperpolarization. But after learning that material, both not only the channels, but the steps of an action potential, we want to make sure we're taking some of the key components about this long-distance signal with us into the next slides. So action potentials belong to the axon. They're an all or nothing signal, or we can call it an all or nothing phenomenon. Graded potentials must reach threshold in order to cause an action potential. Graded potentials and action potentials both start with depolarization. And action potentials are only generated by muscle cells and neurons. Action potentials, unlike graded potentials, do not vary in strength over distance, whereas graded potentials decrease their amplitude or decrease their strength over distance. And the most clinical definition of an action potential is a brief reversal of membrane potential with a total amplitude of 100 millivolts. So we've established resting membrane potential at negative 70 millivolts. So a 100 millivolt difference would be negative 70 to positive 30. And we would see at most of the images that are in this PowerPoint that they peak after depolarization at positive 30 millivolts. We're going to see that action potentials are the way that neurons communicate, how they send signals from one neuron to the other, and that an action potential in an axon of a neuron is generally called a nerve impulse, especially because these nerve impulses travel along bundles of axons, which are called peripheral nerves in the peripheral nervous system. If an organ or a cell or some type of tissue is innervated, it means they have neurons going to it or providing information or signals as a communicative measure. This is a good chart here to review. So generation of the action potential is not only showing you the action potential itself here in blue, it's showing you the permeability of the membrane to sodium and potassium, sodium in yellow, potassium in green, and it's identifying the steps of the action potential. Number 1 being resting membrane potential, 2 depolarization, 3 repolarization, and 4 hyperpolarization. We can see that before we reach threshold, these sodium gates open. The sodium gates opening increase the permeability of the cell membrane to sodium influx, and that we decrease the permeability of the membrane to sodium influx when the sodium gates close when the action potential reaches about positive 30 millivolts. Right after the sodium gates close, the potassium gates open, and they remain open past hyperpolarization. It is the slow closing of the potassium channels that causes hyperpolarization, and it is the ATPase, the sodium-potassium pump, that restores the ionic conditions inside the neuron membrane. Your book also has the step-by-step for the action potential, the permeability, and the channels that open. Remember, the graded potential starts with depolarization after we open a ligand or chemically gated channel, whereas the rest of the potentials, the action potential, it's going to involve voltage gated channels. It will involve sodium and potassium voltage gated channels. The sodium channels, when open, allow an influx of sodium. The potassium channels, when open, allow an efflux of potassium. And when I talked earlier about adjacent segments of the axonal membrane, I was talking about this image. So depolarization doesn't occur all along the axon at once. It occurs in one segment of the axon and then moves to an adjacent segment. Because of how depolarization and repolarization work, the depolarizing signal will not move back towards the axon hillock. Instead, it will move away from the axon hillock. And this is because a depolarized segment will then repolarize, and the nearby adjacent segment that is depolarizing will be unable to move towards the axon hillock because of the repolarizing segment. So we'll have segments that depolarize, repolarize, and then reset to resting, and this will prevent the signal from moving backwards and only moving forwards. So in response to a signal, the soma end of the axon is going to become depolarized. The depolarization spreads down the axon. Meanwhile, the first part of the membrane repolarizes. And because sodium channels are inactivated and additional potassium channels have opened, the membrane cannot depolarize again. So we'll see that the action potential continues to travel down the axon towards the axon terminals and does not perform retrograde movement, only anterograde towards the axon terminals. I recommend watching this video. I'll make sure they're posted on Blackboard with these video lectures. This is the A&P Flicks Neurophysiology Part 2, Generation of an Action Potential. Also the A&P Flicks Neurophysiology Part 3, Propagation of an Action Potential. And we're going to stop there for this part of the lecture. Next, we'll pick up on coding for stimulus intensity.